Everyone can you hear me all right on Zoom? Okay. Okay, sorry. I'm Chen Li. I'm a fourth year PhD student in DCMB in Dr. Joshua Welch's lab. Today's topic is multi-omic single cell velocity models, epigenome transcriptome interactions, and improves cell fate prediction. This is the official publication title, slightly different than the one I posted before, but means the same thing. Today I will go through the multi-value paper recently published. I will go through the background, methods, results, and then the discussion. If time allows, I will show a short multi-value tutorial. Single cell technology has gone through rapid development in the past few years. Nowadays, in a single batch, people can sequence tens of thousands of cells at the same time. So one of the popular platform is 10x genomics, like shown on the left. It sequences each single cell with barcodes and got RNA expression or protein sensitivity counts as a result. With so many cells, people are naturally interested in how the cells develop into each other. For example, if one sequence the brain tissue, they want to know if the ones they label the radionuclei is developing into what other cell types, for example, the astrocytes or the neurons. Similarly, for the brain cell, for the blood cells, they want to see if the HSPC cells are developing into other stem cells or the red blood cells. But there's a fundamental limitation of current single cell technology. It is that when the platform does the sequencing, the cells are destroyed. That means we can't track a group of cells over time to see how they develop into other cell types. For example, in the figure shown above, the cell 2 and the cell 1 that have colors are the ones that we only see. But in reality, we can't see the past or the future of those cells. And we can't really tell what their true ancestors and the descendants look like. To solve this problem, there are many computational approaches that uses these snapshots, single time points, to infer the gene expression changes along the development process. This is what we call the trajectory inference algorithms. They usually use the cellular similarity based on the pairwise cellular expression distance and map to a common pseudo-time axis to define the developmental relationship among these cells. Many methods are developed, including several popular ones, such as monoclonal, palantir, and slingshot. Monoclonal, for example, is the very first one that develops the trajectory inference algorithm. It uses a minimum spanning tree, for example, like shown in the figure. But the issue of the traditional trajectory inference algorithm is that they can't predict the directions or rates of cellular transitions because the distance among cells is bidirectional, and there's no earlier or later definition. That means if we give it a root cell, it can find the later cells, but without a root cell definition, the lineages can go either way. The recent developed RNA velocity actually addresses these limitations by using mathematical modeling of the splicing dynamics. Before going into the detail of RNA velocity later on, let me recap a simple biological definition, pre-mRNA splicing. So during the post-transcriptional process, not only the 5' cap and the 3' polyadenylation were added. One important step is that the introns of the pre-mRNA are spliced out, and resulting mature spliced mRNA for use as a protein template. This is the splicing. This is the mRNA splicing. And the definition of velocity here is that the distance equals the speed times the time. X equals VT, as we learned from physics classes earlier. In a more formal definition, in calculus, we can also say dx over dt equals v. And similarly, in the definition of RNA velocity, we say that the change of mRNA expression equals the mRNA velocity multiplied by the time. Because we have two modalities this time, we have both the unspliced velocity and the spliced velocity, U and S. The definition of RNA velocity is given by the first publication about RNA velocity by Lamino in 2018. It is defined as the first derivative of spliced mRNA S. There are several parameters that can affect the final mRNA abundance. All these are based on the biological process. For example, the transcription rate alpha. This tells how fast the pre-mRNA, the unspliced pre-mRNA is produced. And then the spliced rate beta tells how fast the unspliced RNA is changed into spliced RNA. And then finally, the degradation rate gamma, it tells how fast the spliced RNA level drops. That the whole process is summarized in the figure on the left. We see that when the transcript rate is above 0, like shown on the right, the unspliced count goes up as expected. Then going through the splicing, the spliced count also goes up. In this simplified model, the transcript rate has only two values. One is a positive value for the induction state. This induction state means that the transcription is actively happening. And when transcription terminates, the transcript rate becomes 0. Let's look at the actual ordinary differential equations used in the RNA velocity method. The velocity is v equals dx over dt, like said before. For the unspliced velocity, du over dt, it is the transcript rate minus a splicing rate multiplied by the current unspliced count. And for the spliced velocity, it is defined as the splicing rate multiplied by the current unspliced count minus the degradation rate times the current spliced count. These two formulas essentially summarizes the whole splicing process. If we solve the ODE solutions and plot the unspliced count against the spliced count, we get a phase portrait like shown on the right. It has a classic oval shape. Above the above the diagonal line is the induction state, where transcription is actively happening, and below the dotted line is the repression state, where transcription has terminated. The basic principle is transcriptional dynamics can be inferred by RNA splicing kinetics. Like shown on the diagram to the right, we can see that the root cells and the directors are well defined this time, unlike in the traditional trajectory inference methods. Uh, there are actually two types of RNA velocity models. One is termed the steady state model. This is first developed by the first author of RNA velocity model. Later on, it is improved into a dynamic model called the IC value by Walker Bergen. Because multi-value essentially depends on the dynamic model, just like IC value, I will omit the steady state model, a simpler one, and explain this one in more detail. In this dynamic model, the analytical solutions to the ODEs are obtained. Solving the two ODEs, we got U equals something and then S equals something, both depending on some initial values. Here, because those are the ordinary differential equations, they now depend on T. This is what we call a latent time. It's just another term for pseudo time, but use the frequently parallel velocity. In the dynamic model, the difference compared to the previous steady state model is that all the cells this time are used to optimize the rate parameters that I shown earlier. Compared to the steady state model, where only the top and the lower quantile cells are used. You may see that we are not using T here in the analytical solutions. We use tau because there are actually two states, one induction and one reversion, and these two states have different initial values, so that we use tau to denote the data state. Actually, recently there are even more advanced single cell measurements called the single cell multi-omic measurements. This technology can quantify the protein accessibility and the gene expression from the same set of cells. Not only this time not only RNA sequence. This allows us to see the relationship between the protein and the gene expression. With this new technology, we seek to answer several questions that are previously unsolvable. For example, how do epigenome and the transcriptome change so that the cells can develop into each other, and which modality changes first? If there is a time lag between changes, how long are they? And when do both modalities go up or down? To take advantage of this new multi-omic technology, we developed multi-value as an extension to the classic RNA velocity model. It models the chromatin dynamics as an upstream process to the splicing dynamics used in RNA velocity. Here, we see that on the left, we added two equations, one for chromatin opening and one for chromatin closing. These two equations affect the downstream processes such as extraction, splicing, and degradation along the way. Let's look closely at the new equation. This time, dC over dt is defined as the Kc. Kc is a chromatin state indicator. It is a 1 for chromatin opening state and a 0 for chromatin closing state. Alpha C is the chromatin opening or closing rate. And then C here is the chromatin accessibility as measured in the ataxic count. It's unclear. Just look at the equation, but what it looks like in plot is that it essentially goes to 1 or 0 exponentially, depending on the current state, like shown on the right. We propose this formula both with biological reasoning as well as empirical definition. if we plot the latent, if we plot the chromatin sensitivity along the latent time inferred by only RNA velocity, we see that the chromatin sensitivity goes up and down in a similar pattern as exponential functions, like shown on the bottom. These are 6 example genes that we plotted. Now the biological reasoning for why we choose exponential functions that during the initial impulse of chromatin opening or closing signals, the changes is very quick, but eventually when it becomes nearly open, fully open or fully closing, due to the physical size of nucleosomes, the rate of changes will slow down. The benefit of simultaneously modeling the chromatin and RNA is that we can study their temporal relationship. They are not necessarily always coupled, meaning that when one modality goes up, the other doesn't necessarily follows closely. We think there are 2 possible models, 2 possible orders. We both will start with the chromatin priming phase. This is when the chromatin starts to open, but the RNA stays at 0. Reason that it's unlikely for RNA to be produced on a locus that is closed. So chromatin has to open first. Then both model will go into the coupled induction phase, where both the chromatin and the RNA levels go up. Now the difference is when either modality slows down or drops. For model 1, the chromatin closing precedes the transcription repression state, and the order is reversed for model 2. And finally, both model will reach a stably repressed phase where both modality would drop. This model is this modeling is used for each gene. Each gene will have their own models. In reality, some of the steps, some of the phases may not be observed. We give 2 definitions. First is priming. This is when the chromatin opens before the transcription initiates. There's a time lag between when the transcription starts and the chromatin opens. Decoupling is defined if there's a time lag between the chromatin closing and the transcriptional repression. Like shown on the left, the top one is showing the priming phase, where the chromatin already goes up, but the unspliced count stays at 0. And for the decoupling at the bottom, the chromatin start to drop first, and then unspliced count follows. This is actually only for model 1. For model 2, the order will be reversed. On the right, I'm showing the classic phase portrait for RNA velocity, U versus S. And the diagonal line is the steady state, where this is used in the steady state model. I will not explain that. When adding in the chromatin, we essentially expand the process into 4 states: the priming, the coupled on, decoupling, and the coupled off states. This is also only for model 1. For model 2, the decoupling order will be reversed for chromatin and the RNA. Also on the right, this time I'm showing the chromatin versus unspliced, but then U versus S. It's more difficult to see it in 2D actually, because this time the actual data is 3D. It's chromatin unspliced and spliced, and the simulation result for model 1 and model 2 genes look like the figures here. The difference here is mainly that the chromatin closest state and the transcriptional termination state order is reversed. For the left, chromatin closest first, but on the right, the transcriptional terminates first. Transcription terminates first. The goal here is like other optimization algorithms. So we want to fit a fast trajectory to the observations and solve the rate parameters. We assume that the other observations are IID and the residuals are normally distributed along the true ODE solution. And we assume diagonal covariance in all three coordinates. That means that it forms a trivariant Gaussian along the proposed ODE solution. Here the ODE prediction is F, and then the distribution is given by a trivariant Gaussian. Now to maximize the negative local likelihood, we would maximize the MSE. If we just look at the term at the end. Here I'm borrowing the figure from IC value. This is for RNA velocity only, but it tells a cool story about how the curve is fitted. Essentially, it's similar to linear regression, but it's fitting two ODE solutions to the observed data. This is for each gene. So all these models are for each gene. The actual fitted process is that we now use a list of uniformly spaced time points to represent the actual time. The ODE solution, as I said before, not only depends on the rate parameters, but also depend on the time. We not only don't know the ODE solution, but also we don't know the time either. So these two values need to be inferred step by step and simultaneously. We choose EM also, expectation maximization algorithm, to iteratively optimize the rate factors and the time assignments. So for the expectation step, first the time is assigned to each observation based on the current best ODE parameters, and in the maximization step, the ODE parameters are again optimized based on the given time assignments to each cell. In reality, most times we also observe a partial trajectory for each gene, rather than a complete trajectory that composes of both the induction state and the reversion state. Some of genes only has an induction state, for example. Multi-value uses certain algorithms to predetermine the pattern that speed up the computation. There are two important downstream analyses. Both are based on the dynamical model developed by IC value. The first one is velocity stream plot. This is usually visualized on the UMAP embeddings of the RNA matrix. This summarizes the cell transitions based on the splice velocity and the expression of all genes. And this tells the predicted lineages among cells. The cell transition matrix here is based on cosine similarity. The intuition for this one is that when two cells are expected to be ancestors and descendants, the spliced velocity will agree with the difference between their spliced count. That means that the direction and the splice, the difference will match. Another important analysis is a global latent time. When we fit each gene separately, we get a time for each cell for each gene. But in the end, we want to see a global trend of all the genes so that we can tell the differentiation relationship among those cells across all the genes. I will show the actual examples right now. The first data set we tested is from the 10x Genomics ENT mouse brain data set. Is a public data set on their website. This is based on the cell ranger ARC1, published in two years before. We see that incorporating the chrome gene accessibility and the gene expression improves the accuracy of velocity. Here, the cell type are labeled on the UMAP coordinates, and the expected direction goes from the radionuclide into the astrocyte direction as well as the neuronal direction. The latent time, if we look at the latent time, for example, the darkest point is where we predicted the root cell to be, and as the time increases, the cell type becomes more mature. But if we look at only the RNA velocity result from ICVelo, we see that there are backflows in the upper layer, and there's an unexpected inward flow in ependymal cells. But in contrast, the multi-velo predicts the velocity streams very accurately and agrees with the biological expectations. In this slide, I show an intuitive benefit of including chrome gene accessibility. If we plot the two marker genes for some of the cell types, UMC1, UMC and TLE4, we see that in the phase portrait of U and S, other than the cell type, they are marked. The other cell types all clumped together, and we can't see the details inside those cell types. But if we expand into the chrome gene direction, we see that all the cell types are now well separated. If we model the chrome gene accessibility along with both the RNA expressions, then we can better resolve the temporal relationship of those cells. This one is also benefit of including chrome gene. In a previous review paper for RNA velocity, it's shown that if we slowly increase the transfer rate, the fitted trajectory for induction phase will become more strict, becomes more linear. For this example gene, CDH13 in the mouse brain data set, we observe a similar pattern. For ICVelo, RNA-only result has a very large curvature that is not fitting the data well. But if we include the chrome as well, in multi-velo result, the trajectory is fitting the cells very well in both the RNA phase portrait as well as the chrome versus unspliced phase portrait. In the chrome versus unspliced phase portrait, we see that the chrome increases. That means that the transfer rate this time is jointly defined jointly modeled as the transcription rate multiplied by the current coding sensitivity, alpha times C will increase just as the proposed model. In this data set, we also see clear examples of model 1 and model 2 genes. SATB2 is a model 1 gene. The pointed arrow indicates where the coding closes. This time, in this gene, the coding closes before the transcription regression happens, which is denoted by diamond. But for model 2, the color here is the coding sensitivity. We can see that the coding closing date prediction is very accurate. It is just inside the highest coding sensitivity region. Also true for Greer 2, the coding closing point is accurately modeled within the highest coding sensitivity region. But this time, like in model 1, the coding sensitivity drops after the transcription regression happens, which is denoted by the cross. And plotting the colors with cell types, we see the phase portraits of these two genes show accurate fitting result. Some other analysis for this data set, we see that the model 2 genes achieved the highest spliced expression earlier in later time than model 2 genes, the model 1 genes. For the figures on the right, I'm showing the maximum spliced expression along the latent time axis across the cell types. You see that there are a lot more genes in model 2 that have an earlier spliced expression. We can also summarize the type of genes into a pie chart. There are 4 types of genes possible. For the partial trajectories, we can have election-only genes and regression-only genes. But for the complete trajectory, we have 2 different decoupling patterns, so that there are model 1 and model 2 genes. We see that model 1 is usually the most common. This time, model 2 gene is the second most popular, and followed by induction, then the regression. These figures here show intuitive result of decoupling patterns. On the top, first let me explain the colors. So the red one is the primed phase, the orange is coupled on, then followed by the decoupled green phase, then the blue one is the coupled off. For each gene, any of the 4 states can happen, but they have to be in order. And on the bottom, I'm showing the state assignment for 3 genes, Robo 2 on the first row, Greer 2 on the second row, and Green 2B on the third row. For Robo 2, this gene is model 1 gene. As I said before, for model 1 gene, coding closes before transcription terminates. This is shown on the UMAP plot. If we plot the coding sensitivity and unspliced expressions, you see that when coding level drops, the unspliced expression is still going up. That means that there's a decoupling region, and I'm labeling as a circle. The multi-value model predicts the red region to be the decoupling region, and it agrees with what we would expect. On the right, I'm plotting the dynamic plot for each gene. This means that so this is the time assignment of each cell inside each gene, and the y-axis is the three modality levels: chromatin, unspliced, and spliced. We see that when the chromatin starts to drop in the green phase, the unspliced and the spliced stay at a relatively high level. You may wonder why the unspliced spliced are not increasing. It's because the ataxic is actually an upstream process of the unspliced. It can already determine if the major effect is increasing or decreasing for the RA for the RA counts. For GRIA2, this is a model 2 gene. For model 2 gene, the chromatin sensitivity increases even after the transcription has stopped. Like shown on the third UMAP, the region here labeled as green is the decoupling region predicted by the model, and agrees with what we would expect. Similarly, we can tell from the dynamic plot, the GRIA2 ataxic count still increases when the unspliced and spliced drops in the green phase. Green 2B is either one. It is induction only gene. It only has an induction phase where both the RA and the chromatin increases. It has a clear priming phase. In this circular region labeled as red. Red is the predicted primed phase. The GRIA2 ataxic level increases, but the unspliced and the spliced count stayed at 0. In this data set, we observe a clear cascade of state transitions of 4 states along the neuronal clusters. If we summarize summarize all the genes under the cell states in those genes. We can also plot the predicted state intervals for each gene. This is summarized in the figure on the left. The primed interval, coupled on interval, coupled interval, and the coupled off interval for each gene, which we observe that it is usually shorter for the decoupled intervals. I mean, decoupled is now here is defined as both the primed and decoupled intervals. Those are unmatched intervals. But for the coupled intervals, those are longer, coupled on and coupled off. This is expected because in general, we still expect that the chromatin flexibility and the gene expression to be highly correlated. In this model, we actually models the chromatin closing rate and the chromatin opening rate as 2 separate rate parameters. But we observe that the ratio of the closing rate and the opening rate is a very close one in the end. That means that we can essentially model them as a similar rate. The second data set we tested is the share-seq mouse hair follicle data set. Again, the multi-value predicts accurate lineages for this data set. The PEC1 and PEC2 cells are diverging into RS and the hair shaft as well as the medulla. Also, the length time agrees with the multi-value velocity stream predictions, and says that the terminal states, terminal cell types have a later time prediction. It is very messy in IC value are only prediction. In contrast, we see that this iron velocity result actually agrees very well with pseudo time based trajectory inverse methods. This is the Platea result shown on the right. This is taken from the share sequence paper. For this data set, we see a lot more induction only genes and fewer model 2 genes. We observe one very clear chromatin priming gene in this data set. It's the WNT3. This gene, if we look at the UMAP plot for the three modalities, for the ataxic count, the level goes up very early on in the lineage I label. And then the unspliced and the unspliced and the spliced count follows later on. We used another algorithm called dynamic time warping to compute the time lag between the three modalities. This algorithm non-linearly warps two time series to maximize their similarity. Essentially, it tries to match two time series by expanding some of the time point and shrinking the other time point. And then if we look at the figure on the right, I used the 20 discrete beams for the time assignment as input to the DTW algorithm. And we see that the dotted lines align the predicted matched time points for the two modalities, and there's a time lag between the time points. It tries to match, and we can compute this. We can easily compute this time lag from this algorithm. For the first figure on the right, it is the chromatin and the spliced expressions. We see that there's a very large lag between the two curves. The second figure shows the unspliced and the spliced count. There's a smaller decoupling region between the two curves. If we plot the delta t between the two modalities above, is that the C and S lag is always above the U and S lag. That means that the chromatin and the unspliced lag is always larger than the unspliced and spliced lag. Again, on the bottom, I'm showing the dynamic plot for the three modalities, C, U, and S plotted against the time assignment for this gene. We observe a clear pattern of priming along the way. C, we look at C and U or U and S. The third data set we tested is the hematopoietic stem and projected cells, HSPC. This is the blood cell. Again, using the chromatin information, we observe a more accurate fitting result. Here, the cell types are labeled, and all the cell types are here are actually projector cells of some kind. None of them are terminally stable cell types. For iron velocity analysis, we don't expect that the terminal analysis, terminal cell types to have a good analysis result because they are not differentiating anymore. Here on the left, the predicted direction goes from HIC into all other cell types. some of major cell types are TMP, granulocyte, erythrocyte, and platelet. But for IC value result, the stream plot is very strange. It doesn't start from the HSC and doesn't end at the terminal cell types. In this data set, again, we observe a very similar gene type distributions, like the mouse brain. And again, the switch time interval for the 4 states agreed with the mouse brain as well, with the coupled intervals longer than the decoupled ones. We found that the model 2 gene is are enriched for cell cycle in terms of the geo analysis. Here I'm showing the 3 cell cycle genes for 3 lineages on the UMAP, subsetting for only those lineages, myeloid lineages, erythroid, and the platelet. We see that the ataxic seems to always go up to the end, but the spliced count only expressed in intermediate cell types. This is a model 2 pattern. In this data set, we saw 5 very clear priming genes towards each of the terminal cell types. On the left, I'm plotting the dynamic plot for the 3 modalities for the 5 genes. Just like before, we see that there are certain regions that the coding has already increased, but the unspliced stayed at 0. Besides the absolute expressions for the ataxic and unspliced splice, we can also plot the velocity instead, because this is actually a mathematical modeling. It has the values of DC over DT, DS over DT, for example. If we look at the velocity result, we see the besides the terminal cell types, like at the end, the earlier stem cell populations have increasing RA velocity trend. That means these cells have the potential to differentiate into other cells. But when it reaches the terminal cell type, this velocity drops rapidly, and it's not differentiating anymore. And lastly, on the right, I'm showing the classic phase portrait for the 5 genes. This genes only have induction only pattern. The last data set that we tested is the fetal human brain data set. Just like the mouse brain data set, we see very good prediction in the human. The root cell is predicted to be a circulating population next to radioglial cells, and then it goes into the erythrocyte direction to the OPCs and to the neuronal lineages up to the upper layer and the deep layer. The living time of prediction agrees well with the velocity stream plot, where the cycling population is the earliest time point. IC value, again, there are some more backflows along the lineages that we don't want. Just like mouse brain, again, we see some genes belonging to the model 1 pattern and the model 2 pattern. Robo 2 is a clear model 1 gene. The highest chromatin sensitivity precedes the highest unspliced expression. Then for the MAC2C is a model 2 gene where the coding accessibility follows the highest unspliced expression. And then similarly, the cell type or distribution is plotted. For this data set, we did a more extensive analysis using the latent time inferred from a multi-value. If we assume that the latent time is the true time, we can use it to find the time lag between the RNA expression of a transfer factor and is downstream of binding site accessibilities. We would expect that the expression of transfer factor would first increase, then the accessibility of its downstream binding site increases. And this is confirmed in the 4 genes that we shown here. All those are classic neuronal TFs. Here the magenta is the gene expression, and the yellow ones are the downstream binding site accessibilities. You see that the accessibility always is always later than the maximum gene expression. And we can also confirm it using the UMAP expressions. The highest gene expression appears very early, but the motif accessibility is really quite late. Another analysis is to see if a single nucleotide polymorphism SNPs and they are associated genes. Here I'm showing the maximum SNP accessibility and the maximum gene expression of its linked gene on the UMAP. So the x-axis is the maximum SNP accessibility, and the y-axis is the time of the maximum RNA expression minus the maximum SNP accessibility. That means if the value is positive, RNA is expressed later than the SNP, than the accessible SNP. And if it's close to 0, then they match very well. We only observe 3 islands of the 757 SNPs we found associated with the highly variable genes in this data set. That makes sense because usually for a SNP to affect the gene later on, it needs to be accessible before the gene is expressed, or at least it is closely coupled to the gene expression. For the 3 islands, I plotted some of the example UMAPs on the right. That's the end of result. Some discussions. We see that multi-value can accurately recover cell lineages and quantify the lengths of priming decoupling intervals of the chromatin accessibility and the gene expression. We assume that there are 2 types of gene expression models, model 1 and model 2 gene, and we found good examples of both models in the data set we tested. There are still some limitations in the multi-value algorithm because it's very similar to S-value. Each gene here is treated separately, but we really want to see a global trend that matches all the genes at the same time. rather than treating each one separately and later on aggregate them. Because there can be an inconsistency when treating individual genes. There are more complicated scenarios possible than the two proposed models. For example, multiple reduction repression cycles in the protein or the RNA. Also, it cannot handle multi-lineages dynamics, such as genes with multiple reduction trajectories in different cell types. How this work will be further expanded by members of our lab, Chen. So stay tuned for the final result. I want to thank my mentor, Dr. Welch, and Maria from Dr. Cassie Collins' lab for generating the HSPC dataset. I want to thank the funding sources. Please check out our GitHub. And the paper is recently published in Nature Biotechnology. And if you are interested, please see that. There's only 5 minutes left. I will just explain the input data, and then you can check out the demo for a step-by-step usage tutorial. The major requirements are the output of 10x result. This includes the feature linkage that links the chromatin peaks to genes. Because in this data set, we are using a gene-centric approach. All the peaks need to be aggregated to each gene first, and this file is for that. And then the similar to all the other 10x result, we have the barcode features and the matrix for RNA-seq and the ATAC-seq expressions. We have peak annotations. For RNA velocity, at least for now, it requires a pre-processing step that smooths the expressions among neighbors. This is to solve the dropout issue and makes the gene expressions smoother among the neighbors, so that the trajectory can be fitted well. In this approach, for RNA, we use the similar approach as value that finds KN matrix using only RNA. But for ATAC-seq, we instead uses the Seurat weighted nearest neighbor approach. This considers not only the RNA but also the ATAC-seq. And then the Velocyto is used Velocyto is a method for RNA velocity as well. It is solely used to quantify the unspliced and the spliced count. One way I want to see the RNA velocity is that it is really a method that extends the single modality data set into a multi-modality data set by treating the unspliced and spliced data set separately. And then you can install with PIP or contact and import using Python as a multi-value. Thank you very much for listening, and I'm happy to take any questions.
We are about out of time. If you're online, you have a question, you can put it into Slack chat or Zoom chat. If you're in the room and you've got a quick question, we can go ahead and ask, or we probably can't take 2 minutes today.
So maybe I missed this. Are you filtering genes that you're looking at and then their associated peaks for this?
Yes.
How so?
Yeah. So for RNA expression analysis, there's a filtering step called the highly variable gene selection. Chooses only the ones that with a top variable. Yeah. And for ATAC-seq is similar.
Okay.
Just two quick questions. So first of all, from your some of your slides, it seems that your results is quite different from results from SD velocity.
Yeah.
So have you run some like more quantitative check to see how far apart the two results are? This is the first question. And the second one is this is sounds a very refined work. So have you consider maybe not you maybe be changed in the future? Would you integrate the special information to this pipeline?
Good question. Both questions are very good. The first one, so we haven't developed any specific quantitative comparison for the velocity string plot differences, because currently the results are quite empirical. We can look at the cell types and tell if the transitions make sense or not. But for later analysis, for more complicated models later on, we will develop a method for that. And for the spatial analysis, that's a very good question. That's what we are thinking about, and we'll work on that soon. Thank you very much.
The RNA velocity is just a remarkable idea, and it's incredible that it works so well. And your addition to it to incorporate the chromatin seems like a sort of a natural increase in realism for it. But I guess I'm sort of a two-part question. One is, as you increase the model complexity, especially with like ODE modeling, they become stiff and difficult to identify. So I'm curious if you could comment on sort of the practical challenges of fitting sort of more flexible models, these types of data. And I guess sort of the second part is I can sort of dream up additional complexities that you're not handling. And I'm curious how sort of where do you draw the line, and how do you test if you're overfitting the data? And like what are sort of your general ideas about how do you sort of detect if you've sort of gone too far and you're sort of fitting the noise that it won't generalize better?
Thank you for the question. So for this method is already much more complicated than the original R velocity. I think the original one was with 6 parameters, and this one extended to 10 or 12, I forgot, with a different state switching time, as well as the continuing with parameters, and the free time is a lot longer as well. But because we proposed a simple exponential function that integrates well with the previous RNA velocity models, the results are just as stable as the level optimization result. But if I guess you are right that if we want to extend it to even more complicated models, it can result in non-stable parameter result. That will be very difficult to beat. And then for the second question, I guess there are more complicated formulas for the biological process, such as the master chemical equations that depends on tens of parameters. I don't think that it would work well in terms of the single cell analysis, at least not with a simple optimization algorithm from some type. Using deep learning may solve for such problems, and our lab is also investigating into that.
That doesn't address overfitting.
Oh,
so how do you know that you aren't overfitting data?
That's a good question. Guess for now it's not the issue yet. For the ODE parameter fitting is a purging, and if the real parameters are optimized, then there's not yet overfitting issue yet. But for, I guess for more complicated models, there may be overfitting issues.
So you haven't, there's no way to check, or you haven't checked
No, not
for
yet.
overfitting at all.
We haven't checked this matrix yet, yeah, but we can discuss later.
So it is after the hour, so I think we might need to wrap up, so take our. Anyone who